Hi, this is the PowerPoint on molecular and genomic epidemiology. So I'm not going to read all the slides. I'll just try to highlight what I think is most important from each slide. There are going to be a number of slides in this PowerPoint that talk about that relationship or interrelationship between conditions that are affected by environmental exposure and also those that correlate with a high genetic susceptibility. The fact is it's not purely one or the other. They interrelate with each other. The simplest science is certainly the science of genetics, and you probably studied this in your 8th grade biology class. Genetics is related to what happens when there is an influence from a single gene. An example of this, though, that is part of our, especially those of us who work in pediatrics, is trisomy 21, where a single gene, the 21st chromosome, has an extra repeat in it. So instead of 2 with a repeat, it becomes 3, which is why it's called trisomy 21. The simplest science is certainly the science of genetics, and you probably studied this in your 8th grade biology class. Genetics is related to what happens when there is an influence from a single gene. An example of this, though, that is part of our, especially those of us who work in pediatrics, is trisomy 21, where a single gene, the 21st chromosome, has an extra repeat in it. So instead of 2 with a repeat, it becomes 3, which is why it's called trisomy 21. As the science developed, it evolved into the science of genomics, which is looking for the interaction of all of a person's complement of genes and how that is impacted by the environment and other factors. So there's a good YouTube video link below that talks about what is a genome. And I would encourage you to open that up and look at it so that you understand what a person's genome represents. People's running out and getting genetic tests has become quite the rage, and probably most of us do it for curiosity reasons, but we also are understanding increasingly the impact of our inherited genetic susceptibility to certain diseases. Genes not only cause disease, but they will affect disease susceptibility and resistance, prognosis and progression, responses to illness, and their treatments. So it is very important. Earlier in this course, we introduced the discipline of epidemiology, and we mentioned at that time that there were specialty fields of interest within the broader field of epidemiology, and certainly genetic epidemiology is one of them. You can look here, it's a map that shows the genetic epidemiology of cystic fibrosis. The countries that are highest in the prevalence of cystic fibrosis are in the green, while those are in the lowest tend to be white. And the point here is that the prevalence is actually increasing in some countries. And so this is something that genetic epidemiologists will be following to try to understand how and why that's happening. As genetic epidemiologists attempt to find out if a particular condition has a strong genetic susceptibility, they follow this pattern of questioning, with each one building on the previous one. So again, the first bullet point refers to this interrelationship between inherited susceptibility and the impact of the environment. Just because somebody inherits a susceptibility does not mean that they will inherit the disease because of that impact of the environment. The second bullet point is sort of important before we look at the next slides, and that's the fact that families have to be studied differently in this field because of the family's inherited genes that they share, as well as the fact that they tend to live in the same environment. You cannot look at the members of the family and study them individually. You need to look at them as family units. In the notes section of this slide, it reminds you as healthcare professionals how important it is to get an accurate and complete family history. So it isn't as simple as just saying you have a family history of cancer. Yeah, on whose side of your family? Yeah, my husband's. Trying to ask the next questions will be very helpful to people who are trying to look at if there's a genetic susceptibility for this condition. This is a great slide, but most of its value is probably in the notes section. As we know, then it's not a simple science, genetic epidemiology, because they have to take into consideration that there are multiple factors influencing the inherited susceptibility and what actually gets displayed in a particular individual's health history. So examples of those kinds of influences include the genetic and genomic element, but also environment, lifestyle, and other factors that tend to run in families. Examples of disorders that are particularly multifactorial are given in this long list in the notes section. This slide takes you back to your understanding of a descriptive case control study. So you should remember, I think by now, that a case control study is used to describe what and where something is happening. And we usually start with the cases of concern. In other words, what is the disease or condition that we want to study? And we compare persons or families with that case to controls that don't have it. So this slide looks at, and we will call the cases probands. And this slide also includes where we could get control families, such as relatives of the proband spouse, because of course there's no blood connection since it is a not a blood relationship. So we've been considering what kind of clustering you might find in a group of families that is not genetic. That was the previous slide. So here's an example of some of those influences. If they work in the same trade, it could be that shared occupation. If they live in the same vicinity, an example would be radon, it's in the soil. Religious preferences, followed by lifestyle choices, tend to run in families that stay within that same religious practice. If a family grows up in the same home or even in the same community, an example of that would be lead and paint, or environmental exposure, such as everyone in the household being exposed to secondhand smoke. We need to rule out these non-genetic factors before we can start to identify whether clustering could be related to genomics. So we've been considering what kind of clustering you might find in a group of families that is not genetic. That was the previous slide. So here's an example of some of those influences. If they work in the same trade, it could be that shared occupation. If they live in the same vicinity, an example would be radon, it's in the soil. Religious preferences, followed by lifestyle choices, tend to run in families that stay within that same religious practice. If a family grows up in the same home or even in the same community, and an example of that would be lead and paint, or environmental exposure, such as everyone in the household being exposed to secondhand smoke. We need to rule out these non-genetic factors before we can start to identify whether clustering could be related to genomics. So we've been considering what kind of clustering you might find in a group of families that is not genetic. That was the previous slide. So here's an example of some of those influences. If they work in the same trade, it could be that shared occupation. If they live in the same vicinity, an example would be radon, it's in the soil. Religious preferences, followed by lifestyle choices, tend to run in families that stay within that same religious practice. If a family grows up in the same home or even in the same community, and an example of that would be lead and paint, or environmental exposure, such as everyone in the household being exposed to secondhand smoke. We need to rule out these non-genetic factors before we can start to identify whether clustering could be related to genomics. So continuing on this vein of thought, if it is looking like we have determined that the non-genetic factors are not of importance, then there might be a strong genetic component. This PowerPoint slides lists some red flags that might help to lead to a conclusion that there is a strong genetic correlation. So for instance, the second bullet point there is there a known genetic predisposition to certain genetic disorders? An example of that is Tay-Sachs disease. We've known for a while that it's rare in the general population, but it is more common in people of the Ashkenazi Jewish heritage than other backgrounds. So mutations of this disease are often found in the people that live within that area, which is Eastern and Central Europe, also sometimes in Quebec, an Amish community in Pennsylvania, and a particular Cajun population in Louisiana, and I don't know why. Genetic epidemiologists often rely on twin studies if they can get them, because they provide a wonderful natural experiment. They are looking at whether the condition has a greater relationship or concordance for monozygotic twins versus dizygotic twins versus no twins. So monozygotic, of course, they share the same zygote, so they're genetically identical. Dizygotic are the same as brother and sister pair, two different eggs, and they look at those things. It's especially important if it is a dichotomous trait. A dichotomous trait is either there or it's not. So for instance, a cleft chin or a dimple. But also if they see some kind of a correlation for continuous traits that have a grade in between its absence or its presence, that might indicate as well that there's a genetic susceptibility for that condition. Another group of studies that can be very helpful is migrant studies. So we're looking at a person's immigration pattern. So let's say that they grew up in a part of the world with a high incidence of multiple sclerosis, such as Canada or Sweden, and they end up moving to a location with a very low risk of multiple sclerosis like Israel. It ends up that if they've spent the majority of their childhood in a high-risk area and then move to a low-risk area, the risk of multiple sclerosis often remains high. And that indicates that there might be a strong environmental component to that disease, might be stronger in terms of environment than it is genetic. So I just mentioned the example of multiple sclerosis as a disease that looks like it has a stronger environmental influence possibly than it does a genetic one. But it is an example of a complex disease for which the genetic influence may be modest, and the environmental factors, so anything outside of the individual, contribute substantially to the risk of the disease. These complex diseases tend to have multiple causes, so they're multifactorial, and they're polygenic, so it's not just a single gene, it is the correlation or the combined effect of a number of genes that affect these complex diseases. So I've spoken about the use of the science of epidemiology to understand genetic variation and how that's impacted as well by the environment. This is just a good reminder to say that there is a purpose to this study, and that is to figure out how genes interact with the environment to influence the disease risk and outcome in a population. So in other words, the focus really is on improving population health, which is what this whole course has been about. So what is the future of this science? One thing that is developing, we're switching a little bit from genetic epidemiology to molecular epidemiology. Molecular epidemiology is looking at even a smaller part of a gene, and that's the molecular marker to establish the association between an exposure and a disease. So as an example here, rather than treat all cases of breast cancer as the same disease, they can use tumor markers to identify potentially more heterogeneous subsets and then to treat them differently. So this also has a possibility of providing early warnings for disease by flagging preclinical effects of exposure. So this is really the future. It leads to things like targeted therapies and individual therapies. It leads to fields such as pharmacogenetics. Important for us to understand that we are trying to focus on how one individual with a condition should be optimally handled or have a different treatment plan than another individual. There's a YouTube video about what a genetic marker is as well there that you might want to open up and look at. So this slide looks at how professionals in the field of public health and those in the field of genetics or genomics or even molecular epidemiology can work together and collaborate to improve the health of our populations. Here are some examples of how that collaborative practice can be helping population health. And many times as we develop in different sciences, the ethical component of that science becomes more and more important. So here are some ethical principles about how we can translate the science of genomics into healthcare. The beginning just reminds us that to do nothing isn't the best approach. In other words, to hide in fear isn't going to help the population. Not to act is to make a decision. Instead, we need to use human rights laws to frame these laws to guide the transformation of genomic science into healthcare. It's important that the community, which are really the stakeholders in this collaboration, isn't it? But the community must be consulted. The response also has to be based on good science. And we need to consider the global consequences of these ethical principles. There's an article in Canvas by Badzig that I would suggest you read. As one example that I put in the notes section, it apparently, people who are submitting their genetic samples for testing, some of those donors are demanding control over what happens to their genetic samples. So what are the ethics about that? How can we write policy related to whether they've got a voice in determining what's happening to their genetic samples? When we're looking at policies, the most important policy was passed in 2008, and that was the GINA Act, or the Genetic Information Non-Discrimination Act. In that act, which the link is actually in the notes section, I'd really encourage you to open it up and look at it. It's not that long, but the first part of it is non-discrimination when a person is looking for health insurance. And the second part of it is prohibiting employment discrimination on the basis of genetic information. Certainly, we've come a long way since the very beginning, when early in our country, there were actually laws that forced sterilization of people who were presumed to have genetic defects. Just a terrible, terrible history. And then we moved on, but even in the early 1970s, we were still mandating genetic screening of all African Americans for sickle cell anemia, leading to discrimination, job discrimination, and unnecessary fear for that population. So this is legislation to prevent that from continuing. This slide encourages the role of nurses in this field. It also mentions future research needs to look at the interaction between genetic susceptibility, environment, lifestyle, and all of those, their impact on susceptibility to chronic illness especially. And then in the notes section, I mentioned an article that is uploaded into Canvas called Precision Public Health and Precision Medicine, 2 peas in a pod. Again, how can public health and medicine collaborate together for the good of the individual? And I list 3 priorities of the US Precision Medicine Initiative. The link in this slide is an hour long. I'm certainly not suggesting you listen to it before you take the exam, but even just hearing the first couple minutes of it, it's just nice to see that a nursing professional has really stepped up to the plate and been very involved in the field of genomics. And just to see what nurses can do and how they can incorporate it into practice, or maybe develop it as a specialty of their own near the end of their career. The CDC puts out this scan called the Genomics and Health Weekly Scan. It's got a lot of important information on it. I would suggest you open up, click on the link, open it up, look at the A to Z list, find something that catches your interest, and then read it for, a minute or so, just to get familiar with what might be important to you in your career that could be found at that CDC site. These last slides are really just not at all related to genomics or genetics, but actually just going back to your final power population health paper project. So I want to remind you that you should consider one national initiative in addition to Healthy People 20 or 21. So as an example there, you could use National Prevention Strategy or CDC Health Impact or something else. Also talk about one local initiative. Now you probably won't find something like what is West Ridge, Chicago doing about diabetes. My point is that you could look for a local hospital, or you could consider what Chicago as a whole is doing by looking at the Healthy Chicago initiatives, which are similar to Healthy People 2020. This slide bullet points a number of things that are really characteristic of professional writing, especially at the graduate level. So please make sure that you read it carefully, that you give thought to organizing the content of your paper, not by source, but by theme. And it should be clear to the reader when you're moving from one theme or concept to another. And you can do that with subject headings as well as summary sentences and topic sentences for the next concept or the next topic you're going to discuss. Make sure your writing is professional, and use the ETS software helps that are within Turnitin. So for instance, you would never say review of literatures, because literatures is not a word. The word literature is plural in itself. And those types of things should be caught by the ETS.